Hello, everyone. It's a great pleasure to be here with you in a new webinar promoted by the Consortium of Communitary Universities of Rio Grande do Sul, Comung, Brazil. I am Viviane Bischoff, the Director of International Relations of University of Vale do Taquari, Univates, an university located in the city of Lajeado, Rio Grande do Sul, Brazil. Univates is an institution really committed with the community where we are inserted, and we offer different programs in all areas of knowledge. We also offer different services and research in areas and special fields like health, innovation, and environment. Today we have a great person here to share his experience and his expertise with us, Professor Ivan Bustamante. Professor Ivan will show his presentation about 20 minutes, and then if you have questions, you can share with us by the chat in the YouTube. Thank you very much for your audience, and Professor Ivan, the stage is yours.
Thank you, Dr. Viviane, for the opportunity to be here. It's my pleasure to talk to this great audience. There are some people who are live right now, but I know that this presentation will be available for later audiences on YouTube. So today's topic will be molecular markers in andrology, a biotechnologic approach. I've been working with this theme for about 20 years, and since when I started my master's, and then during my PhD, I also worked with molecular markers, always focusing on how to use proteins to improve fertility in andrology. So the goal of this presentation today is to present you some data from my lab here at Univates. We call the lab Laboratory of Animal Reproductive Biotechnology. Em português, LABRA, Laboratório de Biotecnologia da Reprodução Animal. And we've been working, the laboratory has about 8 years, and we have been working with several students, master students, PhD students, as well. We're here in Brazil, as Brazilians, people from the university in Brazil know this pretty well. We have a huge army of undergrad students who work with us and help us to make everything happen. It's a system a little bit different from other countries, but it's a real Brazilian innovation. So what we're going to discuss today, our first stop, we'll start this discussion with andrology and its importance for human and veterinary medicine. I will present to you briefly the current status of fertility, both in man and domestic animals. Then I will present you some strategies, biotechnology strategies, to improve male fertility, and finally what we have done here at my lab. So first of all about Univates. Univates, it's a university here in the southernmost state of Brazil, Rio Grande do Sul. We have a total number of students about 10,000. And we have around 7,000 undergrad students and almost 700 grad students, including master students and PhD students. We have also more than 900 employees and almost 500 teachers, instructors, and professors. So it's a very good staff, qualified staff that work in this beautiful university, as you can see here in those pictures. I invite you everyone to visit here someday, as soon as pandemic is over. And to show you where we are, so we are about 1 hour and a half by car and by bus from Porto Alegre, the capital of the Rio Grande do Sul state. And it's a very, very nice trip by car, by bus, and the region here is pretty beautiful. As soon as you get the opportunity to visit us, you will see how beautiful this place is, as all the Rio Grande do Sul state. So Univats has graduate courses in biotechnology, my main area here at the university, with focus in agri-food biotechnology and healthcare biotechnology. So the students in these courses are expected to be able to create and spread scientific and technical knowledge of biotechnology in areas of food production and human and animal health, integrating also social, environmental, and economic perspective. Our goal in the university is we always have this focus in everything that we do here at Univatis, with the special focus and the environment and the economic perspective, especially in this region where the university is located. So let's talk about andrology. So for many of you that are not used to the topic, andrology is of great importance in human and veterinary medicine. When we're talking about human health, so man health and fertility is of key importance in human society, especially if we're talking about the maintenance of a steady growth or balance of the population. So economically, we can see several regions around the globe that are downsizing or decreasing, and this brings some economic issues that the governments have to focus on. Also, the men's health are totally related to the generation of new healthy individuals, and this is related to genetics and epigenetics factors. So this is something that we have to also take care of. We must consider the well-being, especially the happiness of individuals and couples, because the distress of not having a condition of have a baby, it's something that it affects the well-being of several people around the globe. And also, we have to comment the significant costs for the public health systems who have to deal with these diseases related to andrology. When we talk about animal fertility, the focus is a bit different, because animal fertility is totally related to the production systems like meat and dairy. So we cannot have meat in the supermarket or meated byproducts as well, dairy and cheese and chocolates as well, if we don't have new animals to make the whole systems turn the wheel. So this is something that we have to keep in mind. So we have to always have new animals coming up to keep the systems flowing. So and that's why I put animal fertility as the cornerstone of animal production systems. And of course, I have to mention the wildlife preservation programs. This is something that's really important internationally, because we are facing the extinction or the threatening of extinction of several species, animal species around the globe, and several research groups are struggling to find new protocols and techniques to maintain, to keep those species alive, if we can say it this way. So especially when we talk about preserving semen from males of endangered species, this is something really important. In the human medicine, andrology deals directly with the causes and consequences of man-impaired fertility. And this is something that I will show you some data about this. And despite clinical and surgical treatments can't be applied, we're still dealing with unknown causes of man infertility or subfertility. The andrology fields relies on several other areas. It's a multi area field. For example, we need andrological support. We need lab support for semen analysis and processing, and of course, the overall health of the individual, including psychological. I will show you some data here about this is a quite old paper published in 1992, where we could see already a reduction in the number of spermatozoa in the ejaculates of men from the 30s to the 90s. So this decrease of semen quality is something that a lot of papers have been showing in the last decades. And however, we have to consider that many of those papers, sorry, many of those papers were published in Europe, in the U.S., where this reduction is often seen more drastically, as you can see in those graphs. The B graph here this lower graphs here show data from South America, so you can see that this reduction is not so big. So we have to consider many things for this issue. So this data in China from 2001 to 2015, we also can see a reduction in total sperm count and progressive cells in the sample. So we can assume that indeed the sperm quality is decreasing. And this recent paper published from some researchers from São Paulo back in 2015 showed also two cohorts of men. It's a very big number, and they showed a significant reduction in sperm concentration and total sperm concentration and sperm morphology. So if you look at that normal morphology, they were expecting something like 4.6% of cells with normal morphology, and now it's about 2.7. So it's almost a half. So this is pretty concerning. And I am a veterinarian, so when I work with animals, especially with bovine and swine, our parameters here, the seminal traits that we analyze here, we expect a minimum 70% of normal sperm morphology. So in man, it's around 5. This is pretty bad indeed. So as I mentioned, the current status in animals is totally different from man. So that's why I put this picture here to show it. We live in a paradise. When we got, if I analyze a fresh semen from a bull, I expect a minimum 90% of cells swimming like babies. Well, babies don't swim, but I think you got the message, and it's totally different from man. And there's several ways to explain this. We have to think that for domestic animals, we are always focused in improve fertility data. So because higher fertility, it's equal to more profits. So the more puppies, the more piglets, the more foals, the more babies we get in our farms, in our production systems, the more money we get. So this is a high selective pressure that especially bovine, ovine, and swine have to select only males and females with high fertile indexes. So if we have any fertility problem with a cow, with a sow, we just take this individual off the herd. That's how it works. So we just keep the animals with higher fertility indexes. This is a bit different from equine, from horses and pets like dogs and cats, because this is a little bit more problematic because the selective pressure is based on the beauty of the animal or the performance. For example, a horse will only serve as a sire, as a stallion will only have semen collections uh, after after its performance in races and other contests. So first we have to evaluate the animal, but the evaluation of the animal is based on their early years as an athlete, and then you will have to check if it can reproduce accordingly. And that's not the case sometimes because we select animals here for other things than fertility. And wild animals, again, it's a different scenario because the thing is that despite the animals have lots of similarities, we have to consider some specific differences involving physiology, anatomy, and some biochemical process in the reproductive tissues. So sometimes you have to you work with an almost distinct species, and you don't have a specific tested protocol to preserve the semen or the embryo of the species. So you have to use a cow's protocol or a mare's protocol, and so it's really complicated. And that's why it's really challenging, but it's a fantastic field to work with. So based on this, what we have discussing so far, biotechnology provides a lot of opportunities to work in the male fertility, because using biotechnology, we have been developing new media for semen, oocyte, and embryo processing. We have been developing new equipment and protocols to cryopreserve cells. We are searching for new molecules that could improve cells' viability and fertilizing potential. And something that came from the 90s, the omics approach, the use of the omics are getting more and more advanced and more high throughput, and we now can identify in a very easier way uh, how to modulate cell events that occur during fertilization. So using genomics, proteomics, metabolomics, and lipidomics, and several other omics that are coming in the next years. And my lab has a special focus on proteomics. So when we discuss here the search for molecular markers, we are focusing here in proteomics. It's the area of science that works with the search for proteins associated with physiological and pathological events. So what have we been doing here at my lab at Univac? So we use proteomics of semen to identify proteins associated to reproductive traits, such high fertility. So when we compare the proteins present in the semen of individuals with high fertility with lower and against lower fertility, we are able to see the differences. And if we know the role of those proteins found differentially present in those samples, we can infer which biochemical and molecular processes are being up or down regulated or totally shut down, and then we can find new ways or test new ways interfere, to change this situation. So that's why we search for animals with high fertility, low fertility. We compare animals with different seminal qualities, and also something that's really important for us in the animal science field is the sperm resistance to cryopreservation. This is not a huge problem for human reproduction because human semen, for example, cryopreserves pretty well. We don't have too much trouble freezing human semen. However, different species, domestic species, have different, how can I say, different responses to cooling and to freezing. So, and those are techniques that are really important because, for example, we have a huge part of our herds, bovine herds, swine herds, now equine herds, who relies on artificial insemination. So to cryopreserve by cooling or by freezing is the best way to ship the semen from one farm to another, from a stud to another, to spread the genetics, the best genetics, to different herds and provide a continuous improvement of the genetic quality of these herds, of these production systems. So now I will show you some briefly, very briefly, because each paper could be, I could talk about it 1 hour about each paper. So I will show you quickly what our data, what we have been what we have been publishing the last year. So, for example, in this paper published this year in Terrazonology, an important journal in the animal reproduction field, we compared. Our goal was to test how the seminal plasma, the liquid where the spermatozoa come in when they're ejaculated, if the proteins in the seminal plasma, if they change when those bowls are subjected to scrotal insulation. Scrotal insulation is a technique that wants to mimic a thermic stress, a heat stress. Brangus bulls are bulls that is a crossing between European cattle and Indian cattle. So we are always trying to find animals that can resist better in the Brazilian environment. So to raise cattle here in Brazil, animals must be very tough to resist in our summer without reducing their weight or the meat production or the milk production. So our goal was to test how which proteins were changing during this stress. So we identified 5 proteins related to several processes inside the testicles that could be used as a marker. So we saw that TIMP2, spermatoxin, and those free BSP proteins, bind of sperm proteins, that were that they were decreased during this stress. So based on this data, we could identify what was happening in the testicles during this stress, the heat stress, induced heat stress, and now we can figure out new ways to perhaps pharmacologically minimize this stress in the hottest days. So this is some of the possibilities that those studies bring to us. Changing the species, now we will talk a little bit more about the swine, because the swine production here in Rio Grande do Sul is very, very important, especially here where Univates is located. And we published in the last year some very interesting data comparing different samples of ejaculated from boars. And firstly, we identified the proteins that were associated with the sperm quality. So we found that some proteins were highly expressed in the seminal plasma of animals with better seminal quality, and this could be used as a marker to help the selection of ejaculates. And also we tested if those proteins were also associated with the sperm resistance to cooling at 17 Celsius degrees. This is very important because the swine production really depends on artificial insemination, and normally we have boar studs where the semen are collected and processed and shipped to different farms. 1 day, 2 days, 3 days trip until it will be used in artificial insemination. So it's important to understand why there are some semen samples that resist pretty well the preservation, and there are some samples that just are completely are in a completely bad shape after the preservation. So we tested for the proteins, and we also find proteins that could be used as a marker for higher resistance to cooling. And now this paper was in cooling at 70 degrees. And in this other paper that was really recently published with some colleagues from the Federal University of Rio Grande do Sul and PUC also, and we tested, we made a slight change in the selection. First, we selected only animals that had a very good semen quality after preservation at 17 degrees, and then those animals were tested for semen preservation at 5 degrees. And why we are reducing the temperature of the semen preservation? Because there is the European market, the European Union is asking to reduce the use of antibiotics in semen, something that we have to use when we preserve at 70 Celsius. So our goal here was to try if those animals have semen that could resist at 5 degrees, and at 5 degrees we don't have to use antibiotics. So we run the proteomic we run the proteomic tests and experiments, and we also find proteins that were present only in animals with higher resistance, and other proteins that were found in lower resistance in the lower resistant ejaculated. So again, we have new evidence that we can use as a molecular marker to select animals with improved resistance to 5 Celsius. Again, changing a little bit of changing the species. Now this work also was recently published in Andrologia, a German journal in the field, an important journal in the field, and we investigated a protein that I'm really interested in, the protein disulfide isomerase. It's a protein involved with the quality control of other proteins. So these proteins acts fixing damaged proteins. So it plays a very important role in different biological systems. So our goal here was to check if the PDI-A1 was involved in seminal quality and it was different expressed in the epididymis of foals of different ages. So our goal, we measure this protein in very young foals and medium age foals and then older foals, almost adults, to check if there was an association with puberty. So this was a very nice work, and we are still working on this to check if those proteins could be used as a molecular marker for fertility and seminal quality in the stallion. um, After, as I told you, I want to be quick, and I would like to show you now some data that this is something really new, something it's very authentic because it's an animal model for man secondary hypogonadism, and this is an idea that I had several years ago, and because here at in Brazil, we have a we have we normally we castrate uh, pigs using a product that acts as a vaccine against a hormone called GnRH. So this hormone is very important to induce the pituitary gland to secrete LH and FSH, which are responsible for the normal functional development of the testicles. So since we in this animal model we use a antibody against these hormones, we block this hormone axis, inhibiting the secretion of LH and FH, and we cause a condition called secondary hypogonadism. And this is really interesting. We do this in farms here because we have to sell the animals without the testicles or vaccinated. This is pretty common here. So I saw this as an opportunity to use those animals vaccinated against GnRH as a model to study the secondary hypogonadism in men, because this is an important cause of infertility in men in several countries, as I mentioned, induced by an improper secretion of GnRH, FSH, and LH. However, to study this in men, it's not really easy because we have several limitations to study this condition and its consequences in the testicular and epididymal function. You cannot ask for a patient in a clinic to provide his testicles or epididymis to investigate it. It's an organ that it's not a good thing to get a biopsy or something. So you need an animal model to study that, and then make this translational to translate the data to try to get the most of this data to investigate in a proper way in human. So our goal was to identify putative candidates for diagnostic and treatment purposes. So our first the first paper I would like to show you was we using also proteomics. We identified several proteins that were differently expressed in the epididymis of boars that were immunized against GnRH. So this first graph here shows that about 300, you normally find 300 proteins in the epididymis of normal boars, control boars, healthy individuals. But when we vaccinated, we have more than the double of the number. So, and more than 400 exclusive proteins that wasn't there if the animals were not vaccinated. So there really something's going on, changing the whole scenario of protein scenario in the epididymis. So our goal was to identify different relationship between proteins and possible explanations to identify why the blood blocking of the hormones could totally impair the function of the testicles and epididymis. So this paper here, we focused on the epididymal fluid, and in this other paper here, recently published, we assessed the proteome of the spermatozoa from the epididymis. So we also, differently from the epididymal fluid, we didn't see a huge increase in the number of proteins. It was a very mild change. However, more than 100 proteins were exclusive found in those animals with secondary induced secondary hypogonadism, and also we had to trace each protein to check the role of each proteins in the testes and epididymal physiology to try to understand why those changes would impair the fertility in man. How could those proteins change the way the spermatozoa works or survives, not only inside the male reproductive tract, but also after ejaculation inside the uterus and the oviducts. So this field is a little bit tricky because we have huge amounts of data. We found more than 1,000 proteins, and it's like a puzzle that you have to put together, put the pieces together. It's not that simple. So our goal here was to find those proteins that were differently expressed, and those proteins that were and how the changes in those proteins could impair several steps involved in the fertilization process. And not only high throughput proteomics, we've been using also several bioinformatics and gene expression studies to investigate more specific candidates like beta defensins and also the PDI that I have already mentioned. We have been studying more specific proteins that have higher potential to be good markers for fertility. So what can we do with all this information? Our goal is to develop new diagnostic methods using more specific markers for specific conditions. Then we have to compare properly each condition to find those markers, and also have the statistical support to be sure that those markers are really related to the conditions we are working on. Also, better comprehend the pathologies, allowing the discovery of new treatments. This is really important. Also, our goal is to improve semen processing protocols for assisted reproductive techniques like in vitro fertilization, using recombinant proteins, because one of those proteins, if they're really special, we can try to create a biofactory to produce exclusively these proteins in a bacterial system in order to add or supplement the sperm with a missing protein, for example. And finally, to provide a better animal selection based on protein markers in the semen, and to develop molecular shortcuts to improve the preservation of genetic material of endangered species. This is really important because we don't have time to investigating all the endangered species we know. So if the more we know about the species that we were working on, maybe we can find shortcuts to apply to pandas, to apply to other endangered cats like puma and etc. and etc. So finally, I would like to thank you for the audience for listening to watching my presentation. Would like to thank all the agencies that fund our work here FAPERGS, Univates, CNPq CAPS, especially for the scholarships, our international collaborators, and also, of course, the Brazilian collaborators, and my team that have been working with me the last 8-9 years here, making those data available for you that I show you today. So that's it for now. I'm here. I'm available for questions if you have some.
Okay. Thank you, Professor Ivan, for your presentation. This is not my field of study, of course. I'm far for that, but I think it's a great moment to person know more about this topic. We have some questions that the audience sent by the chat of YouTube, and I am also have some questions that people sent to my WhatsApp. So the first question, Professor Camille Granada. Ivan, there are several evidences that the women's pregnancy after 40 years improves significantly the probability of a baby born with different diseases. And women's pregnancy with old men, the probability to born a baby with disease also improve?
Yes. There are several papers on this topic, and of course, in animals, this is not a problem because the animals don't reproduction to the older age. But there are in man, yes, especially related to oxidative stress. I didn't mention here because there are so many things to talk about this topic, but our society now, and this might explain why in developed countries we have more problems with fertility in men than in underdeveloped countries, because the exposure to chemicals, to endocrine disruptors and pollutants, and this is something that we have to consider. And also drinking and smoking. This is something that increases the oxidative damage to the DNA in the germ cells in the testicles, and there are several data I can show you later that shows that after 40 years old, the DNA damage in the spermatozoa is pretty big, and this damage could induce a abnormal embryo development and causes pregnancy failure or a newborn with genetic problems. And I'm just talking about genetics. We also have the epigenetic factors that are involved too. So this is something that, yes, older men, and this is something that our society is changing. Many men now are marrying for the second, third time, and they have 50 years old and they want to have babies, and this is something that they have to consider.
Okay, thank you. Another question sent by Professor Raul Sperotto. Ivan, which is the animal model that has the reproductive system most similar to the human one in terms of anatomy, physiology, and proteomics? And to which extent the findings in an animal model can be extrapolated to the human reproductive system?
Yeah, that's the 1 million question, because the more we know, the more we notice the differences, and this is for sure. Of course, we still have to use animal models to investigate human diseases. We have to, because there's no way you can develop a drug or a new treatment without only testing in cell cultures. you need a system with immunological environment and so on and so on. So I think for each organ, for each tissue, for each disease, there are better models, but a perfect model don't exist. There are several papers showing that data generated in mice or rats are not good enough to extrapolate, as you said, into human physiology and pathology as well. However, that's why we have to test to develop a drug or other treatment. We have to test in 1, 2, 3 species to be sure that we can use in human. However, there are several drugs that failed. That they were pretty good in rats, in mice, in pig, and then in monkeys and different apes, but then in human was not good. So for each condition, there are the specialists agree that there is a better model, but there is not one model that fits all. The pig model is pretty good. That's why, for example, our colleagues from Universidade Federal de Pelotas, they are working on pig models, deleting genes from the pig, trying to make it more to look to immunologically to be immunologically closer to human, and that's what they have been doing the last years. So I don't know if I answer your question, but
okay. I have received actually 2 questions directed to my WhatsApp, because we have some audience abroad, and some partners, institutions send. I will try to do the 2 questions in one. Professor Ivan, about these papers you showed us, did you have international collaborators? And are you looking for new international partners now?
yeah, of course. And if you, I think as a researcher, I learned this with my advisors. And if you want to have good quality results, if you want to get the most from your data, if you want to learn new techniques, new approaches, you must collaborate with international institutes, universities, and companies. This is in science. Collaboration is the basic. You have always to collaborate, and not only here in Brazil. I have collaborators here. It's very important. Colleagues that we meet in congresses, in meetings, but also in the abroad. I've been working with a researcher from Durham University, and some of those proteomic data were generated in a collaboration with the Scripps Research Institute in California. And now we are just started a new project with the university in Italy, in Bologna, with the veterinary faculty there, to investigate proteins associated in resistance in swine semen cryopreservation. That's the pandemic is slowing everything down, but we are working on it. And last week, I just started a new collaboration with Uruguay, and we are here in Rio Grande do Sul. Uruguay is closer than São Paulo and Rio, so why not to internationally collaborate? So it's good for me as a researcher. It's fantastic for my students if they have the opportunity to interact with international partners, and that's one of the greatest things in science, that you can interact with other people, not only online like we're doing right now, but also in the laboratory or in meetings and of course in the parties after a whole day in a meeting watching lectures. We have a party or we go to bars, pubs, and this is one of the good things that I learned to do in science.
Of course you have. And we have a message here. It's not a question, but Professor Cristiana from University of Santa Cruz do Sul says, greetings from the International Office of University of Santa Cruz do Sul, UNISC. Congratulations, Dr. Ivan, for this presentation and important research. Okay.
Thank you.
So, if you have no more questions, I would like to thank you, Professor Ivan, for your presentation, for being available to join us in this project. And thank you very much for all the audience. As Professor Ivan has mentioned, this webinar will be available for all people interested to see again or to share with friends, with colleagues, and also will be available for the new audience. Thank you very much for all the colleagues from Univates for the structure to offer this webinar. And Professor Ivan, if you want to say something else.
Well, again, I would like to thank the opportunity to show our data here, and we are open to new collaborations here in Brazil, abroad, and we are very eager to do more things in this field. And of course, if we have a technique or a protocol that we do that could fit and help in other areas that normally happens, feel free to get in touch with me in my email. My email is in the presentation. If you want to check it out, it's not hard to find me online.
Okay. So thank you very much and see you around the world.